Well, hello and welcome to Bio 211. This is our first lab and it is the blood lab. So this is a screenshot of our handouts for this lab. Now online under content on D2L, there will be the student handouts. I highly recommend that you might not have to print them out now that everything's pretty much online, but that is where everything that we go over is listed. I'm not going to, unless I say it specifically, I'm not going to go over things that aren't on this list. And there are a lot of things I will be telling you can cross off the list. But this is the one for today's lab. I put it on this particular one. It might not be on many of the other ones, but I wanted to put this on here. Again, things are going to be changing as we go along. This is kind of still unusual. And so anyway, this is our lab schedule for today. So what we're looking at is blood. And the first thing we're going to talk about is the components of blood. Now, one of the components is this thing called formed elements. Now, formed elements, we're going to look at, the formed elements are going to be cells. There's going to be 3 different types of cells. But the other thing is called plasma. And again, I don't have a fancy computer where I can have a pen to write on. And so I'm having to do looks like a little kid with crayons in here. So plasma is the other part. And so the next slide, I want to kind of look at what this means. So this is a picture from your book that divides the blood into various parts. You got whole blood, and then it gets divided up into, like we say, formed elements and plasma. Now, the plasma is going to have a lot of different things in it. The main constituent of plasma is water. We will look at plasma in lecture, but I do want you to know blood is made up of formed elements and plasma. Now, our lab today is mainly going to be about these formed elements. Now, this lists the formed elements. There's 3 of them, 3 different types of cells that are in here, or cell types. And they're called platelets, red blood cells, and white blood cells. And you can see red blood cells make up the majority of the formed elements by far. But you will also need to know these by their scientific name. Now, on the lab handouts, they have the scientific name. Erythrocytes are red blood cells, leukocytes are white blood cells, and thrombocytes are platelets. So the first part of this lab, we're going to look at, like I said, the divisions here. Then we're going to look at white blood cells, and then we're going to talk about red blood cells or erythrocytes. So what I want you to know is how blood, whole blood, is looked at, and how doctors and medical providers look at blood and how they use it. The first thing happens is if you go to the doctor, they're going to take a sample of your blood, and they'll put it in a vial, and they'll send it through this little thing called a centrifuge and spin it around, and blood will separate, and there's going to be this little area here. Now, we're not going to worry about the name Buffy coat and this plug, because those are the 3 parts of the formed elements. So basically, it's formed elements that are going to be heavier and they sink to the bottom. They're going to be roughly 45% of whole blood. And then we have plasma, which plasma is 55% of whole blood. And so when the doctor does this and looks at it, they can kind of tell the health because of this ratio. If all of a sudden we have less formed elements, that ends up being a catch-all term that's going to be referred to as anemia. If there's more, that's going to have a different effect on things. But that's what we look at. That's called a hematocrit. Again, you're not going to have to know it here in lab, but this is something we're going to look at in lecture. It's called a hematocrit, but you definitely need to know formed elements and plasma are the 2 components of whole blood. And the formed elements are going to be the 3 blood cells. We're going to have erythrocytes, which are called red blood cells, which is the majority of it. Leukocytes, which are white blood cells, and thrombocytes, which are platelets. So again, that goes back to this. This is the scientific name for those 3 things that are formed elements. And you need to know how to use both of them. Again, I'll probably have questions that will ask you one or the other, and sometimes I'll have a question looking for a formed element, and I'll be specific and say it needs to be the scientific name. So if I ask you what's the most common formed element and I need the scientific name and you say red blood cells, that's going to be wrong. All right. So what we're going to look at now ends up going to these. We're going to come back to the erythrocytes, the red blood cells, and blood typing. But what we're going to look at now are the leukocytes, the white blood cells. And they are divided up into two categories, granulocytes and agranulocytes. Now, I am big on knowing words. Anytime you hear or see an A in front of something, that means it is without. So a granulocyte versus an agranulocyte. The agranulocyte literally means without granules. And so we're going to look at, there's 5 different types of leukocytes. And again, they're divided up into granulocytes and agranulocytes. And you can see them down here as well. This lists them in their groups. These are going to be what we're going to call the granulocytes, and these are agranulocytes. Now, before we look at some of these pictures, I'm going to describe what you're looking for. With these pictures, I want you to be able to recognize some certain characteristics of these. Now, one thing I do want to say, an agranulocyte actually does have granules. You just can't see them. They're not the prominent feature of it. So when we start looking at these 5, I'm going to point out things that are unique for each one of these cells. Things that you should hone in on because they're going to be the big billboard signs that say, this is me. So as we go through, again, I want to focus on some things. I'll tell you some certain things about each one of the cells to give you ideas about what they do. And you'll need to know certain things, and I definitely will let you know and key you in on what you need to know. But a lot of this, again, is going to be mainly for lecture. So the white blood cells, as it says, the leukocytes are divided up. This gives a number, which we will come back to, a percentage. In lecture, we're going to look at this. This is called, when a healthcare provider looks at these, this is called a differential. And if any of these percentages is out of its range, it'll give the clinician an idea of what's going on. Now, the first one we're going to look at is up here is the neutrophil. The neutrophil is the most common of all of the formed elements, or of the leukocytes, of all the white blood cells. So if I'm looking at this picture, this is an actual picture from our slides. All of these little guys here are the red blood cells, or as it says up here, erythrocytes. But we've got this one that's a little bigger than normal right here, and it's got this weird look. It almost looks like if you're looking at this picture looks like if you're looking up at the moon and you see that face in the moon. But this is the next one is the picture I want you to kind of hone in on. This is a picture, a classic picture of a neutrophil. Now, a neutrophil does have granules. You can see all these little dots here and there, and that is fine to kind of hone in on it. But the key to the neutrophil is this weird shaped multi-lobed nucleus. The original name for a neutrophil was a polymorphonucleotide, which everybody kind of gets all freaked out because how long it is, but it's actually a really good, really descriptive name. Poly means more than one, morph means weird shaped, nucleo means nucleus. So it meant that there were more than one weird shaped lobe of the nucleus. To me, this looks like if you went to a carnival as a kid and you got a little balloon animal and you thought it was cool and you left it up on your desk and it started to inflate or deflate and it starts looking all weird. That's what I think this looks like. But that is the key to the neutrophil. It is going to have multi-lobes, at least 3 on any picture I'm going to give you, of weird shaped nucleus. So of a weird shaped nucleus. Now again, these are the erythrocytes or red blood cells that are all around it. A couple of things I do want you to know about these. They are the most common type of leukocyte. 60% of all of the white blood cells are these neutrophils. They are by far the most common. So 6 out of every 10 are going to be a neutrophil. The other thing I want you to know about it, and I'm going to draw a little, I don't know if anybody here is old enough to remember who Pac-Man is, but I'm going to draw a little wedge shape here because that's what these guys are. These are little Pac-Men. They're called a phagocytic cell. What their job is to go through and engulf foreign invaders and dissolve them and get rid of them. That's what phagocytosis means. But they're a phagocytic cell, and you need to know that. So the next one is an eosinophil. Again, I think that these pictures are kind of tough to really use. So I stick with this. Now, there's 3 granulocytes. We just looked at a neutrophil, which has granules, but they're not the star of the show. The next 2, the granules, are definitely the star of the show. So this one's called an eosinophil, and its granules are these really dark red marble looking granules that are in here. And you can see the nucleus, and it actually does have a weird multi-lobe nucleus, not quite as much as the neutrophil. However, that's not the star of the show. The star of the show are these granules, these red granules. Now, I have a lot of different ways that when I was a student or whatever, having to remember and learn things. And so there's a lot of little sayings that I've learned that go with these. They are personal to me. Some of them make sense. Some of them don't make sense. Some of them are just rhymes to help you remember things. This one was kind of personal, and it is a little bit religious in nature. So I hope that you understand. I'm not pushing any of this on you. I just want to get you to understand how to come up with a way to remember this. So when I was in school, see, these have very red granules. The eosinophil has really, really, really bright red granules. So I could remember that the red granules were eosinophils because I grew up as a Catholic or a Christian, and I remember being taught that the blood of Jesus covered my sins. So it's called an eosinophil. So I could remember that the red granules in the blood was the eosinophil, to try and tie those two together. If that works for you, great. If it doesn't, no big deal. I'm not trying to push anything on you, but just an easy way to remember that. Now, one of the things we're going to learn about the eosinophil when we are in lecture is that this is really big in allergies. If you suffer from allergies, your eosinophils kind of go a little haywire. So that's the eosinophil. The next one, again, this is still in the granulocytes, is called a basophil. Now, one of the things, hopefully you remember this or were told this in 210, which again, the way things were last semester, if you took 210 last semester, there was a lot of things that probably didn't get communicated as well. So just to let you know, anytime you hear basal or baso, it means bottom or lowest. This is called a basophil because it is the least common of all the white blood cells. Now the basophil, we're going to see on this next slide, has very dark purple blue granules. I've had students describe them both as blue or as purple, and I've said it either way, but they're really dark blue purple granules. Again, it looks like a bag of purple marbles or blue marbles. Now, again, it has the multi-lobe nucleus in here, but you can't really see that. So that's not the star of the show. The star of the show are these very dark purple blue granules. And again, this is called the basophil. It is the least common of all the white blood cells. Now, I do want you to know there are 2 types of granules here. The first one, or one of the types, has something called histamine in it. I don't know if, and I'm probably going to misspell this. I probably shouldn't even try here. Histamine. I don't know if, everybody's probably heard of it. You might not know what histamine is. Histamine is an inflammatory compound. It causes vasodilation of blood vessels. The other one, and I'm not going to try to write this out, is called heparin. Heparin is the average person on the street would call heparin a blood thinner. That's not necessarily a great word if you're in the science field. It's really called an anticoagulant. It stops blood from clotting abnormally. So what this cell does is, let's say I have a splinter that makes it through my skin. I'm going to have bacteria from that splinter get into the dermis, which can get into the bloodstream and cause havoc. So what this cell does is, when it comes there, it drops off these histamine and heparin granules, and it causes the blood vessels to vasodilate, which means I'm going to get more blood to the area, carrying more white blood cells, particularly neutrophils, trying to engulf any particles that are bad. And then, because I don't want the blood to clot before it gets to the actual injury site, the heparin makes sure that as that new blood comes in, it doesn't clot before it is needed to. So I do want you to know basophils have histamine and heparin in them. But again, it is the blue, the dark purple, dark blue cell. So those are the three types of granulocytes. Again, neutrophil, multi-lobed nucleus, weird shaped nucleus, eosinophil, red granules, basophils, blue granules. So now we're going to look at the agranulocyte. The very first one is called a lymphocyte, and as you can probably guess, it's going to be associated with the lymphatic system. If you've ever heard of T cells or B cells, which are, what we consider our immune system, they are both lymphocytes, and we're going to look at this when we get into the lymphatic chapter. The lymphocytes are really amazing cells. Outside of a neuron, they're probably the most amazing cell in the body. They can live outside of the circulatory system for decades. They actually learn how to fight specific diseases, and then they hang back as memory cells waiting for our body to get invaded again. We'll talk about that at another time, but I'm going to show you the next picture, which is the better picture of it. So this is, again, the better picture of a lymphocyte. And the thing about a lymphocyte is that it ends up looking like one big purple dot. And it's because it is almost entirely one big nucleus. It is a nucleus with a light blue halo around it. That's kind of what it looks like under the microscope. Now, as I said, these cells are the ones that we specifically classify as immune system cells. And so the immune system, what we think of, like we're going through this whole idea of this virus going out. So if we caught COVID-19, these cells would learn how to unravel it and learn how to attack it. And then once we defeated it, these cells would have certain ones of these hang back as memory cells. And anytime we come into contact with it again, cells would become activated and instantly attack it. That's how our immune system works. And then last of these is called a monocyte. Now, one of the things I want you to know about a monocyte is it can also leave circulation, and when it does, it is called a macrophage. And again, I should have not tried to write this out, but a macrophage, which means big, and phage, which I'm not writing out, means eater. So macrophage is a big eater. So we looked at a neutrophil, which is the most common of the white blood cells. It's a granulocyte, where this one is an agranulocyte. But they're both phagocytic cells, but this is a big one. And you can see that from this picture. The monocyte is the largest of the leukocytes, of the white blood cells. And its key feature is it has a very large nucleus. It's not like the lymphocyte, which takes up the whole cell. And it's not like the neutrophil, which is all weird. It looks like a big lima bean. On a monocyte, it's going to be bigger than half the cell, and it's going to generally be shaped like a big lima bean. Now, again, the thing about this is it's another one of those little Pac-Man cells, these little phagocytic cells that go through. They eat any of the bad guys. They try to engulf them. And the neutrophils, because they're the most common, if I have an injury, they will come in. In abundance. But when these guys come, they're like the bulldozers of the group. If you've ever seen the movie The Nutty Professor, when they go to the buffet and they eat all of the food except the salad, which I always think is pretty funny. But that's what these guys are. When they come in, they go through and they do damage. Now, again, what I wanted to make sure I remind you of, these are called macrophages. And so throughout this semester in lecture, when we're looking at different things, you're going to see, we're going to talk about macrophages in different organs, in the lungs and in different areas. Understand that a macrophage is simply a monocyte that is no longer traveling around inside the blood vessels. Now, this is a little mnemonic to remember kind of the percentages. I'll tell you the honest truth, I'm not going to test you on this, but it is kind of handy, especially if you get into nursing and you're kind of looking at things. There's going to be times when you'll have to kind of understand what a differential is, and this is a very simple mnemonic. And again, mnemonics don't have to mean anything. They just have to have something that triggers your brain to tie it in. And so it is: never let my engine blow 60, 30, 830, right? Never let my engine blow 60, 30, 830, which, again, neutrophil, lymphocyte, monocyte, eosinophil, basophil. These are rough percentages. 60% going to be neutrophils, 30% lymphocytes, 8% monocytes, 3% eosinophils, and as it says here, it's basically 0% are the basophils. They are the bottommost. So the next part of this, we're going to talk about blood typing. So blood typing is a really big deal. Blood typing is how our body will start looking at what is you and what is not. It kind of gets into that idea. And so we're going to look at how different cells, right? So if I draw a red blood cell here, it's going to have something on its surface, generally, called an antigen, all right? The antigen is going to be on the cell surface. Now, we're looking at this through a blood typing lens. Just real quick, I want to make you aware of the fact that the term antigen actually refers to a foreign invader. So if we again COVID-19, if it gets into our system, there are antigens on that virus that are signals to our immune system to attack it. And so in our blood, we're going to have antigens on the cell. And the reason it's called an antigen is because, let's say if this is type A, so this is an A antigen here, that means that B antigens are bad guys to us. So the B antigen acts as an official antigen. That's why it's called that. Now, the other thing is there's going to be floating around in my plasma little things called antibodies. So I'm going to erase this little A here and I'm going to show you what happens. So I've got this little antibody, and the antibody is only looking for one thing. So let's say that this is type A, and I'm going to go over some more stuff with this in a second. So that means that this is an A antigen right here. If I have and you've got to remember these things are called is the first part of the name. So this is an anti-A antibody. So if I know it's named anti-A antibody, it tells me what it doesn't like, right? If let's say I am if I told you, yeah, I like football, but I am anti-Gamecocks, or I'm anti Clemson or whatever. Hope I don't upset anybody with that, but as long as it says anti in front of it, tells you what it doesn't like. So if I've got an antibody called anti-A antibody, it doesn't like A antigens. So what happens is, as soon as my immune system sees this antibody is going to dock onto this antigen. It's going to hijack the cell, and we're going to talk about this a lot more in depth, but I'm just giving you an overview. And it causes the cell surface to become sticky. And the cell will start sticking to all the other cells around it. This is referred to as agglutination. It's an abnormal blood clot that's caused by an antigen-antibody reflex. Now, this is what, I'm giving you the overview here. We're going to talk about it here in a second. I'm going to pull up the next slide to kind of give you some ideas. Now, this is such a weird thing, and I probably should have had its own little special video on this, but I didn't. All right, antigens and antibodies. First, when I am a fetus, when a fetus is growing, right, and forming in the mother's womb, there is an early point where the fetus's blood will become set as to what blood type it is. Now, there's two kind of separate blood types. There's an ABO system, which is what this slide is. And then there's an RH system, which is whether it is positive or negative. And I'm going to look at both of them separate first. But if I was to combine them, there's only 3 antigens. Right? So on the ABO system, which is again what this is, all right, it's called the ABO system of blood typing. In the ABO system, there's only 2 types of antigens. There's an antigen A and an antigen B, and that's it. So as my body is forming, I'm going to go down a distinct pathway, and I'm going to have a certain blood type. And the blood type is going to be named for the antigen or antigens on my cell. So if I have antigen A here, I am type A. If I have antigen B, I am type B. Pretty straightforward. If I have both of them, right, so I've got both A and B here, I am called AB. I am blood type AB. If I have neither, like this one does, then I am O. So those are the 4 types. I have A, I'm type A, and then I have B, which I'm type B. If I have both, I'm AB, and if I have neither, I have none. I'm O, all right? Now, hopefully this is such a simple thing, but it's so easy to mess up. So I'm begging you, make sure you get the foundations first, because if you try to run before you know how to walk, you're going to trip and fall every time. All right. So antigens are on the cell surface. In the ABO system, there's only 2 antigens, an A antigen and a B antigen. All right. A or B. All right. Now I can have both of them, and then I can be AB, or I can have neither and be O, but it's pretty straightforward there. Now, when my body is on its dedicated path, once it is determined what blood type I am, my body will look at the blood cell and figure out whichever antigen is not present is my enemy, and therefore I will make antibodies that want to attack it. So for instance, if I'm type A, that means I have antigen A on my cell surface. All right. Since I do not have B on here, when my body is in, the process of building everything up, it knows since I don't have B, B is my enemy, so I make anti-B antibodies. And remember, I don't know why so many people want to leave off this first part. That's the biggest part of the name, and that's the part of the name that's the clue. If I tell you that there's anti-B antibodies somewhere, you should be able to tell me what antigen they would attack. Anti-B antibodies attack B antigens, right? So if I'm type A, I will have antigen A on my surface and anti-B antibodies in my plasma. If I'm type B, that means I have B antigens on my cell surface. And since I do not have A antigens on this, that means that A is my enemy. So I have anti-A antibodies flowing around in my plasma. So those should be pretty straightforward. Those shouldn't be that tricky. The trick is if you understand the process, then you'll understand why these bottom two are the way they are. So if I've got type AB blood, that means I have both A and B antigens. And so when my body looks at it and says, which one do I not have? And it's like, oh, you got both of them. That means I have no antibodies because I'm not missing either one of them. So if I go over to the next one and I'm type O, I don't have any one of the antigens, right? There's no A and there's no B. Those are not present. So now, since I don't have either one of them, I'm going to have both antibodies, an anti-A and an anti-B antibody. Hopefully that makes sense as we're going through this, all right? So, and if not, please go back and watch that again, just to try and make sure, because I'm telling you, the weird thing on this is if you don't get the foundational parts of this, you're going to mess up on so many things. Make sure, even when you start doing this on quizzes, even if you feel comfortable, do not try to jump ahead. Always sit and think, all right, if I'm type A, what type of antigens are on my cell? A. Which antigen do I not have? I do not have B. So which antibody is in my plasma? It would be anti-B antibodies. All right, this time I've got a B antigen on my cell, so I am type B. I know that I do not have A antigens, so they're my rival, so I'm going to have anti-A antibodies. If I am type AB, that means I have both antigens. And if I have both antigens, that means I'm not missing either one of them, so I have no antibodies. And if I'm type O, that means that I don't have either one of the antigens, and therefore I will have both antibodies. Now, what happens is so let's say I'm type A and I have B blood put into my body, I'm going to have a reaction. And we just talked about it being called agglutination, because this little guy right here, once he gets into my system, these will attack it and cause it to clump up, which we call agglutination. I'm going to show you the next picture of it here. This is what happens. This is agglutination. Right? They stick together. Again, I've got a kind of a weird imagination. I think of it very much like if you've ever seen the movie The Incredibles, where Mr. Incredible is trying to run. He gets caught at the computer and he's trying to run out, and those little sticky balls get him and he can't run any faster, and it finally wears him down. And that's kind of what happens with these. As they start sticking together, they can't move around anymore. And then they will be what we call, they agglutinate. They clump up, and that can be very deadly. So I'm back to this slide. So I want to look at how blood typing works. And actually, I kind of wish I was able to edit this, but I don't know. I don't think I can. So we're going to look at two things on this ABO system. One of them is called recipient, and the other one is called donors. All right. So recipient, if I'm a recipient, and this is where I want you to be selfish on this. Think of it as you. If I'm a recipient, that means I am receiving blood. If I am receiving blood, that means I need to worry about what my body's antibodies are and the antigens of the cells that are coming into my system, because in blood, donating or receiving, all of these get taken out of the plasma of the person that is getting the blood pulled from them. All right. So if I'm receiving blood, I'm not receiving any of their antibodies. I'm just receiving their blood cells, which is important to remember. So if I'm receiving, I've got to look at what antigens are on the cells that I am getting, that are putting into my body, and will my antibodies react to it. So let's just go through a quick little look with this one, and it should be fairly obvious on this one. So I have antigen. I'm type A, so that means, and go through the steps. If I'm type A, that means I have antigen A on my cell. If I'm type A, that means I do not have B, so I have anti-B antibodies. So if I'm a recipient, if I'm type A and I need someone to give me blood, and this is, again, not worried about positive or negative right now, just the ABO system. I'm looking at it to be put into my circulatory system. I have to make sure that my anti-B antibodies do not attack it. So the question is, what do anti-B antibodies attack? And hopefully all of you right now, while you're watching, are going, it's easy. It is anti-B. That means that it attacks B antigens. So if I have anti-B antibodies, that means I cannot receive blood from anyone with an B antigen, right? So I can receive blood from A's, right? I definitely cannot receive blood from type B because they have a B antigen. Can I receive blood from AB? AB has a B antigen. So I cannot receive blood from type AB. Can I receive blood from O? Does O have a B antigen? It does not. So I can receive blood from O. So as far as recipient goes, if I'm an A, I can receive blood from A and I can receive blood from O, right? Now just to do a couple more here, I'll go through these. So if I'm type B, going through the steps, don't try to run too quick. So if I'm type B, that means I have B antigens on my cell. And as far as recipient goes, that's not what I'm worried about. So I've got B antigen, I do not have A, so I have anti-A antibodies. Anti-A antibodies in receiving are what I'm worried about, my antibodies. And if it's anti-A, it tells me very clearly what it does not like. So if I am type B, I cannot put any blood in my system that has an A antigen. So I cannot receive type A because it has an A antigen. Obviously, I can receive from type B. Type AB has a antigen A, so I can't receive from him. But O, there's no antigen on the cell, so O can also be one that I can use. So as type B, I can receive from B and O. I always like to make a joke about that. BO. Anyway, sorry. Now the ones that get a little more complicated, those hopefully you're sitting there saying, wow, that's pretty straightforward. So let's look at blood typing, being a recipient if I am AB. If I am type AB and I need to receive blood, right, I'm worried about, again, my antibodies. If I'm putting blood cells into my body, I am worried about their antigens. And that means I am worried about my antibodies. So if I'm type AB, what antibodies are in my system? I'm going to give you just a second and think, and then I will tell you there are none. There's no antibodies. So I've got nothing that would attack anything. So I can put A into my system because there's no antibodies. I can put B into my system because there's no antibodies. I can put O into my system because there's no antibodies. And of course, I can put AB into my system. So hopefully you see that because it has no antibodies over here, AB can receive blood from anyone. That is why type AB is considered the universal recipient. Now, if we move on and we look at type O, O doesn't have any of the antigens, so that means it has both sets of antibodies, and that can become a problem. If I need to receive blood, I cannot receive blood from anyone with an A antigen or a B antigen. So there's only one person type O can receive blood from, and it is other O's. Now, where receiving blood is all about looking at my antibodies and try to decide if I'm going to, the blood that I'm putting into my body, I got to look at their antigens to see if my antibodies will attack. Donating is the opposite. So if I'm donating, and again, you look at it like it's personal here. Let's say I am type A. That means I'm going to take my blood, don't have to worry about my antibodies. I'm going to take my blood and put it into someone else's system. So if I'm donating, what I'm looking at is my antigen and that's not what I was trying to write, their antibodies. All right, so let's go through this. So if I'm type A and I want to see who I can donate blood to, if I am type A, that means that on my blood cell surface, I have A antigens. Now, I want you to think about this. What antibody does not get along with A antigens? I'm going to stop just for a second or two and let you think about it. Hopefully you said anti-A antibodies. And I'm going to stress again, anti-A antibodies. Anti-A antibodies. You have to remember it is called anti-A. A lot of people want to leave that off for whatever reason. So if I'm looking at this, it's this simple. If I'm type A, the only people that I can donate blood to are people that do not have anti-A antibodies. So can I donate to A? Of course I can, because type A, they won't live if they have anti-A antibodies. Could I take this cell and put it into someone who's type B? Of course not, because they have anti-A antibodies. Could I take this and put it into A? Yep, I could do that because they do not have anti-A antibodies, because they don't have any antibodies. Could I take this cell and put it into type O? Nope, because of the anti-A antibodies. Hopefully that, again, makes sense. So we'll go through this. So type A, if we're looking at, and I want to warn you, do not try to memorize this. Understand how we get there. But just to tell you, type A can donate to A, and it can donate to AB. All right. Now, if I'm type B, that means that I have got B antigens on my cell surface, so I cannot donate to anyone who has anti-B antibodies. And so there's only two that have anti-B antibodies, and that is type A and type O. So B can donate to other Bs, and it can donate to type AB. All right. Because it's as simple as mix and match. As long as you remember to name things correctly, it's as simple as knowing that these two things don't go together. So if I'm type AB and I want to donate my blood to someone, I have both antigen A and antigen B on my cell. That means that I cannot go to anyone that has anti-A or anti-B antibodies, right? So is there anyone on here besides type AB that they could donate to? And the answer is no. If it went here, it would cause agglutination. If it went here, it would cause agglutination. And here, definitely cause agglutination. So finally, we get to type O, which is a fairly interesting type here. So type O, if I want to put my blood cell into someone else's body and I'm type O, I have to worry about the antigens that are on my cell surface and the antibodies in whoever's receiving it. So if I'm going to take this and put it into anyone's body, I don't have any antigens on my cell, so are there any antibodies that would attack it? And the answer to that is no. So O can give blood to anyone. O can give blood to A, to B, to AB, and of course to other O's. That is why O is considered the universal donor. So AB is the universal recipient because it doesn't have any antibodies. O is the universal donor because it doesn't have any antigens. Now again, we'll go over this a little bit in lecture, but hopefully you get that. This will be a couple questions on the test. And then the last thing we're going to really talk about is the RH factor, which is the other component to this. Now, the difference is, and this is a big one, in the RH factor, you either have it or you don't. So, all right, I'm going to draw two cells here, and I'm going to say this one has the RH factor. I'll draw it as an R. So it is positive. That means it has it. The people that don't are negative. And so if you've ever heard of someone being A positive or A negative, the A is telling you in the ABO system which antigens are there. The positive or negative tells you the RH antigen, whether it is there or not. Now, the big deal is neither one of these have antibodies naturally. Neither one is born with an antibody. Now, before we say anything, well, as I get through this, because this is, again, it's relatively straightforward, but it can be kind of confusing. So I want to make sure I get this right. So if I have it in positive, if I don't, I'm negative. Now, if I take this and I put a RH negative cell into an RH positive system, nothing will happen because there's, as far as the RH goes, because there's no, there's nothing on the negative cell. So when we're looking at things and tying in overall blood donating or receiving, negative blood types can go into any blood type. The problem becomes if an RH positive gets into a negative system. Now, in the ABO system, like if this was type A and I put it into a type B system because B would have the anti-A antibodies, as soon as this got in there, agglutination would happen. I'm going to just put AG, right? Now, if this happens because there are no antibodies present at the beginning, this person's system will capture this like it's an invader, start breaking it down to figure out what's going on, and then we'll start making anti-RH antibodies. All right, I'm just going to put R there. Anti-RH antibodies. If the cross-contamination happens a second time, then agglutination will happen. Because by the time that this person makes these anti-RH antibodies, the original cells will be dead and gone. But if it happens again, boom, it's on. Now, this is where a lot of people will hear it because it's big in childbirth. If I am an RH negative woman and I give birth or I become pregnant and I have an RH positive fetus, all right, so What it's showing here is she is Rh negative, and then all of a sudden she's pregnant. The baby is Rh positive. There is a placental boundary here that's going to prevent the mother becoming developing these antibodies and attacking the child. So during this first pregnancy, some of the child's blood will get across, and the mom will develop, or the blood won't come across, and the mom during delivery there will be a contamination. And again, I don't know how many people have had children, but I hope this doesn't upset you. But during childbirth, there is some blood that's involved, and so at that point, when the child is being born at childbirth, then the mother has this first contamination where she will recognize it and then start making these antibodies to fight it. Now, the real problem is if she gets pregnant a second time, almost, I won't say instantly, but very early on, her body will attack this little baby and will abort it. Now, they have drugs now, and especially in developed countries, that's called Rogan. At one point in time when I first started, I called it Rogaine accidentally, and I couldn't figure out why everybody was laughing. But as soon as I caught what I had said, all I could think of is this little Chewbacca baby coming out. But Rogaine is for hair growth. Rogan stops this. And so that is one of the things that happens that they give to pregnant mothers, is Rogan to prevent this from happening. So I hope this makes sense. Now, again, if I put everything together, there would be actually 8 different types. I would have type A negative, which would be like this one that doesn't have the Rh. And then I would have A positive, which would have like an Rh. I'll just draw a little circle here. That would be A positive. Now, if I am A negative, I can give to A positive. But if I'm A positive, I cannot give to A negative, especially twice. All right, and that's how it works. Now, most of the questions I'm going to ask, I'm going to try to separate the ABO system from the Rh system so you don't have to cross that over. But again, once we get into lecture on this part, I'll go over it again, and hopefully it will click. The big thing, and I hope you hear me when I say this, understand the basics of this. The people that have problems try to jump ahead and like memorize a chart, or, okay, if I just memorize who can donate to who and who can receive from who, believe me, that ends up becoming a real problem. If you just understand, because it's simple. In the and the whole scheme of things, there's only 3 antigens. 2 of them are grouped as ABO, in the ABO system. The other one has its own, but there's only 3 antigens, an A, a B, and an Rh. The A and B are grouped together, and there's only 4 types of blood: A, B, AB, and O. And then on all 4, you can either have the Rh or not. So I can have A positive or A negative, B positive or B negative, AB positive, AB negative, O positive, O negative. But at birth, the only antibodies that would be present are anti-A or anti-B antibodies. Those are there to attack their respective antigens. So anti-B attacks B, anti-A attacks A. And it's called agglutination, and it happens instantly on that. On the Rh factor, no one is born with any anti-Rh antibodies. So it would take a cross-contamination twice to have any real effect. And if it happens twice, then you get agglutination, and that can be a big problem. But that's the blood lab. There will be questions at the end of the week that I'm going to have posted that will go over this. That will be the weekly quiz. And so, hopefully we've got this all covered. Take care. Have a great day.